Bismillahirrahmanirrahim. Assalamu alaikum. It's Dr. Maria here. Today I'm going to talk about one of my research article being published in the Journal of Medicinal Chemistry by Bentham Science. The article entitled as Potential Biological Targets Prediction, ADME Profiling, Molecular Docking Studies of Novel Steroidal Compounds which we obtained from the biotransformation of tibolone through the culture of Cunninghemella Blakesleana. We had a drug tibolone which is used in the hormone replacement therapy. We conducted its biotransformation studies by using Cunninghemella Blakesleana culture. We got 6 new metabolites from the co-design studies, and then we used in silico based techniques for finding out its drugability criteria, for finding out its, the pharmacokinetic studies, for conducting its membrane permeability studies, and for predicting profiling of biological targets prediction. We further validated the results and then we conclude very good results. So in the co-design studies, all of the compounds showed excellent drugability criteria according to the Lipinski rule of 5, and all the compounds showed excellent pharmacokinetic properties. None of the compounds showed any inhibition against any isozyme of cytochrome P450 enzyme, so there is no possibility of drug-drug interactions. And 5 of the compounds showed blood-brain barrier permeation, while 1 of the compounds showed human intestinal absorption permeation. All the 6 compounds are perfectly fitted within the pink acceptable radar zone of bioavailability radar, and none of the compound exceed any of the bioavailability radar descriptor, which includes lipophilicity, flexibility, molecular size, polarity, insaturation, and insolubility. So all the compounds are perfectly fitted, and they shown very good bioavailability radar. We use these compounds to predict their potential biological targets prediction studies, and we profile up their studies, and we profile up their properties actually, and we used in silico based software Swiss target prediction for its studies. And very fortunately, we got 5 of the compounds to be active against the transcription factor of glucocorticoid receptor, while the 1 compound showed to be active against the transcription factor of androgen receptor by using on the highest probability for its studies. And very fortunately, we got 5 of the compounds to be active against the transcription factor of glucocorticoid receptor, while the 1 compound showed to be active against the transcription factor of androgen receptor by using on the highest probability by using 2D and 3D similarity search methods with the respective database of CHEMBL, and the obtained results we furthermore validated by using molecular docking studies with the help of site-specific method through AutoDocVina software. The software is really very good and efficient to conduct the molecular docking through site-specific method, and we got good results, and we validated the results. So 5 compounds which were predicted to be active against glucocorticoid receptor with highest binding affinity showed good hydrogen bonding with the active site amino acid residues of the receptor, while 1 of the compounds showed to be active against androgen receptor with highest interactive mode and with highest binding affinity of against the targeted receptor. So in conclusion, and furthermore, we also used Reactome biological bath phase prediction database to find out that how these compounds have the capability to reach at the site of action with the interaction of steroidal hormone dependent proteins. So in conclusion, in silico-based studies always gives us a rational drug designing and drug discovery approach. So it's a very smart strategy to profile up the compounds before conducting any of the experimental studies, because it saves the time, it saves our manpower, it saves the chemicals, it saves ultimately a lot of efforts which we conducted if we do it blindly. So a smart strategy is that we should have to use in silico based studies at first before to go on the experimental process, and we use this smart strategy in our case and we got very good result, and I hope so this article will be a very valuable addition in the scientific community, and I'll suggest you and recommend you to read my article, to cite it and use it in your studies according to your design experiment. Thank you for joining us.